Welcome back, biologists, to part 2 of Chapter 7D. Still on adaptive immunity, but now we're going to focus on the cell-mediated response. So getting stuck straight into it, try to make this one a bit shorter. The learning intention is to learn about the adaptive cell-mediated immunity and the cells that are involved and what their roles are. So to be successful, your goal will be to be able to name the various cells involved in an adaptive immune response and explain the differences between B cells and the various types of T cells, which are cytotoxic, helper, and memory cells. All right, so with your B cells, we've already talked about the fact there are two types, plasma and memory. T cells for your adaptive immune response, you've got cytotoxic, helper, and also memory as well. We'll talk about all of those and what their roles are. So with your study design, we're still focused on the same part, same chapter, same area, so we'll just move past that. So when we looked at the innate immune response, this grayed out section at the top, these were the cells that we talked about. So we talked about eosinophils, mast cells, and their roles in releasing histamines, both in terms of inflammation, and we'll look at that in terms of an allergic response later. We looked at neutrophils as a phagocyte. These two, we looked at macrophages and dendritic cells as your antigen-presenting cells. And then we also looked at natural killer cells, all from the innate immune system. Okay, so all non-specific but important second-line defenses. Here we're focusing on our third line, the adaptive or acquired immune response. Please note acquired and adaptive are interchangeable terms there. And so here you've got cells involved in your cell-mediated response. They're indicated with this little green dot. All right, so your cytotoxic T cell, helper cell, and the T memory cell. Because remember, your B lymphocyte, both plasma and memory cells are involved in creating the humoral immune response, the chemical response, the antibody response. We've already looked at that in part 1. So our main focus is going to be, we're going to touch on these a little bit because remember, T helper cells activate the B cells. So we don't sort of get away scot-free and don't mention them. But our main focus is on the T cells and what they do. So we've already talked about in the last section the features of adaptive immunity. We've talked about why it's specific, the specificity of antibodies, the specificity of T cell receptors that also need to be activated on T helper cells. But what I've highlighted here, the only difference between this slide and when you saw it before, is that I've put memory cells in bold. We're going to focus a little bit about what memory is, what that looks like. So just a reminder that in order to activate your T helper cells, you've got antigen presentation. We've already talked about that in the last video, so I'm going to skip past that, but it's just a reminder of what we've talked about because we are talking about T helper cells and the fact that they play a very important role in activating the cell-mediated immune response as well as the humoral response. And in order to be activated, they've got to see it, see the antigen. They can't see it without it being presented to them. How is it presented? Antigen-presenting cells. Hopefully you remember which cells are your antigen-presenting cells. I just touched on it like 2 slides ago. But do remember this is so important. There's questions about it quite regularly. They're often multiple choice, only worth a mark. But remember, every mark gets you closer to that score you want. So don't give up easy marks. And these are pretty easy marks. The 2 antigen-presenting cells are macrophages and dendritic cells. The main one that is of absolute utmost importance is dendritic cells. They are special because they can actually travel through the lymphatic system, which we look at in the next chapter. And they can actually take the antigen-presenting cells to where the lymphocytes are. I mentioned in the last video that B cells are called B lymphocytes because they're in bone marrow, B for bone marrow. T cells are called T cells because they reside in the thymus. And so dendritic cells can actually travel through the lymphatic system to the thymus to take the antigen to them where they are. So they're important. They often have a specific and special mention both in the study design, but also in exam questions. So what do antigen-presenting cells present the antigens to? You should be yelling the answer at the screen, I hope, or at least saying it loudly in your head. They present it to your T helper cells. And again, we saw this slide in the last video for part 1. Why? Because these T helper cells even do play a part in humoral immunity, which is our focus for the last video. So we're looking at it again, but now we're looking at it with respect to what it does in cell-mediated immunity. So again, just that reminder, because repetition means retention. The antigen is processed by the phagocyte. It presents it on the surface together with its MHC2 marker. The T cell receptor will be highly specific to that antigen. It will bind to the antigen MHC marker complex. It is then activated, and then it can go on and, in the case of humoral immunity, activate B cells, or in the case of cell-mediated immunity, do what we're about to look at now. So T cells are lymphocytes. So they're sometimes called T lymphocytes as well. They are involved in the cell-mediated response. The ones that we're focused on are your helpers, your cytotoxic, and your memory cells. Before we go any further, I've touched on T cell receptors a couple of times, but T cell receptors, or TCRs, are made of 2 polypeptide chains. So remember we talked about antibodies having 4 polypeptide chains? These only have 2. Okay. Like antibodies, they have both variable and constant regions. That generates variability, which creates the specificity. One receptor will respond to one antigen. Antibodies have 2 binding sites. You saw that with that Y shape they've got. T cell receptors only have 1 binding site. That's the key difference in terms of their structure. T cell receptors will not bind directly to antigens. They bind to the fragments presented by antigen-presenting cells. It triggers signal transduction, which results in the T cells proliferating, in other words, dividing, multiplying, making more. Release of cytokines. Those cytokines are chemicals which activate B cells, but they also activate cytotoxicity, okay, which is done by, it will surprise you to learn, a cytotoxic T cell. T helper cells are helpers, as the name suggests. They don't do anything directly themselves. They don't directly eliminate the pathogen. They secrete cytokines and promotes inflammation, which leads to the recruitment of immune cells. Remember, we looked at inflammation in the innate response. Recruitment of phagocytes, recruitment of more immune cells, increase of fluid in the area, all that kind of stuff. They activate macrophages for phagocytosis and B cells, but they also have a role in activating cytotoxic T cells. Cytotoxic T cells recognize and kill foreign, infected, or abnormal cells. Please note what that says there. Foreign cells, infected cells, or abnormal cells. So we're not here talking about it directly killing the pathogen. They kill the cell. They insert something called perforin, which, as the name suggests, it perforates the cell membrane of the cell. It releases a substance called granzyme B, and granzyme B triggers a process called apoptosis. Apoptosis is programmed cell death. So there's a whole bunch of new words and concepts in there. Let's just break it down real simple. Cytotoxic T cells kill cells, your cells, or foreign cells, say from a transplant. They don't kill pathogens. They do that by sticking a hole in the cell membrane, sticking an enzyme in it, which triggers apoptosis. Apoptosis is just a controlled and programmed death of the cell. So it kills the infected, abnormal, or foreign cell. So what do cytotoxic T cells do? All right, so your first step here, you've got your antigen presenting cell. It's going to activate the naive T cell. T helper cells release cytokines which help stimulate this naive T cell to differentiate, similar to B cells. B cells formed plasma cells and memory cells, but here they produce cytotoxic T cells and T memory cells. So those are the 2 other cells that we needed to talk about in the cell-mediated response. So again, let's break down the 3 cell types. They're all involved here. The helper cell just helps by releasing cytokines, chemicals, to stimulate differentiation. The specific naive T cell has specific receptors that have received the signal from the antigen presenting cell. So again, it's specific, highly specific, 1 T cell receptor for 1 antigen. But together with the T helper cell, they're stimulated to produce cytotoxic T cells. Need to go back 1 step there. All right, so your cytotoxic T cells, they will then go on and bind to cells that are presenting their antigen like this, or that are missing MHC markers because they're damaged, things like cancer cells, all right, and virally infected cells. Virally infected cells will often or will lose their MHC marker, all right, or they'll have antigen presented on the surface. The cytotoxic T cell will recognize that, because remember, viruses live inside the T cell. They multiply, or not inside the T cell. They live inside your body cells. They multiply by taking control of your DNA and copying themselves. They do not float around free in the body, and if they do, they're not causing any harm. It's only when they're inside cells. So when the cell is infected, the signal is sent out to the cytotoxic T cells to say, hey, I'm infected, come kill me. It doesn't kill the virus, it kills the cell. By extension, it then kills the virus because the virus can't replicate because the cell's now dead, right? but it's not directly killing it. It's actually killing the host cell. That's really important. A memory T cell is produced after the helper and cytotoxic T cells have been activated. They, again, are antigen specific, and they will respond rapidly on a secondary exposure to the antigen. In other words, you've had it once, it retains a memory. If you get exposed again, you get a much, much faster response. Why? Because you're not going through that process of selection. Remember, you've got to select the right clone, you've got to find the right cytotoxic T cell. That all takes time, a couple of weeks. You've got the memory, it's already been selected. It gets activated, it starts proliferating really quickly, dividing really quickly. You get a much faster response the second time around. Why are T cells important? They are the only response the immune system has to a pathogen inside a cell. All right, so if it's hiding inside a cell, like a virus, T cells are the only things that can do anything about it. B cells won't see them. Right? B cells will not encounter their antigens because the antigens are hiding inside a cell. You won't be able to stimulate antibodies. You won't be able to do anything. All right, so or you won't be able to target the virus directly because it's hiding inside the cell. So even if you make antibodies, it still can't get into the cell to kill the virus. Cytotoxic T cells detect it by the recognition of the MHC marker and an antigen fragment on the cell surface. They go, yep, okay, that's where it is. I need to kill that cell. So what is the mechanism? I've touched on this already. Here's your infected cell in the middle. These guys are healthy cells. The healthy cells are not displaying the antigen. Therefore, the cytotoxic T cell will go, yeah, oh no, this cell's good. Don't need to do anything. But it detects the complex, all right, antigen with MHC, detects the complex. That then triggers the cytotoxic T cell to release perforin. Perforin sticks a hole in the membrane, which then allows it to insert chemicals. The chemical is called granzyme B, and it kills the cell. As it says down the bottom here, it induces that process, cell death, apoptosis. Now, memory, I don't care if it's a B cell. In this case, this is actually an example talking about B cells, or if it's a T cell, but the graph shows you basically what I want you to see. If you have your initial infection, or a vaccination, so you're exposed to the antigen, however you want to talk about it, you're exposed to the antigen is processed, it is presented, it triggers the humoral response, and it triggers the cell-mediated response, in other words, B cells and T cells. But see here, you've got this lag time, all right? This lag time, a couple of weeks. Then you'll get this bump, the primary immune response, it'll make antibodies, it'll clean up the infection, and it retains a memory. So it doesn't need this 2 weeks anymore. It already knows what to do if it sees it again. That's the key. So now you get a natural infection or a second infection. This time your lag time is really brief, but not only that, not only is it brief, but look at the size of the response relative to the first one. Massive response mounted. Why is that important? Well, here, you probably got symptoms. You probably got sick. You finally triggered your response. Your antibodies cleaned everything up. Your cytotoxic T cells cleaned everything up. You got better. But you had a couple of weeks of feeling like crap first. Here, the response is so rapid and so large that it actually will probably mop up the pathogen before it can cause any symptoms more often than not. Or if you do get any, they're only pretty mild. Right? So that's the importance of memory. And it's why quite often, you'll hear people say, oh, you can only get chickenpox once. It's kind of true. It's not 100% true because your immunity can drop over time. But in part, that there is some truth to that but why is that? It's not that you've never seen the chickenpox virus again ever. It's called varicella zoster, if you care. That's the name of the virus. It's not that you've never encountered it again. You probably have. You probably had chickenpox. You might have, a cousin or a family friend or something. Their kids have had chickenpox, but you didn't get it because you've already had it once. Well, why? Because you've retained a memory, and you were exposed to the virus. Sure, it got into you. Yep, got through the barriers, but this response was so overwhelming you didn't get sick. So that's the importance of memory. And again, remember, humoral memory cells produce these antibodies. So that's what this graph is actually showing you. It's the humoral response, the chemical response. T cell memory will trigger cytotoxic T cells in massive numbers that will then kill infected, abnormal, or foreign cells. So that brings us to the end of chapter 7D. In the first part, you should have learned about the humoral immune system. But now what I want you to be able to do is understand the key differences between, skipping over this because we're going to name the cells here, key differences between B cells, which mount a humoral response, a chemical response, and can only do so to pathogens outside of cells, and the different types of T cells and their role. So that, what does the helper cell do? Well, it triggers B cells, but it also triggers cytotoxic T cells. What does the cytotoxic T cell do? inserts perforin for infected, foreign, and damaged cells, and kills them by inserting granzyme, which triggers apoptosis. That's what a cytotoxic cell does. And your memory cell, your memory T cell, what does that do? It retains the memory so that if you encounter that antigen again, the response is rapid, it is specific, and it stops you from getting sick. So hopefully now you can tell me what a B cell does, what antibodies are, what their structure is. Hopefully you can now tell me the different types of T cells, the 3 different types, and what they do, what their roles are, and tell me the difference, importantly, between a humoral adaptive response and a cell-mediated adaptive response. We will absolutely go through this again in class. If you have any questions, we certainly will go through them in class, and I'll explain anything more. Again, I encourage you to make some notes, but in particular, questions. Let's unpack this together. You should be building a summary around this. You should walk in on day one with a summary that's already explained this, maybe not fully because you didn't fully get it, and that's okay. But the bit you don't get is the bit I want to know. I don't want to have to tell you this whole story right from the beginning. If I do, we're going to lose time. It's going to be a real challenge for us to embed this properly for you. So learn this as best you can with gaps, sure, and then let's fill those gaps. All right. I look forward to hearing the questions you've got. I look forward to getting feedback on how useful this was, and I look forward to seeing you guys back at school next week. Thank you for taking the time to watch these screencasts, and I hope they helped.